How do we go from a genotype to a phenotype? That's what the Central Dogma is trying to explain. The Central Dogma says that information exists in DNA and it flows into RNA, and that an information RNA is used to make protein. And why do we call it the dogma? Because virtually in every living organism that gene expression going from genotype to phenotype that has been studied, these are the steps that move an organism from genotype to phenotype. So we're going to talk about the different steps that are involved in this central dogma. Now, gene expression in prokaryotes and eukaryotes are different, even though the series of steps going from DNA to RNA to protein are the same, but in details they are different. So in prokaryotes, where there are no membrane-bound organelles, and there is a genetic material that's sitting in the cytoplasm, then there are genes on it. The genes on the DNA are going to be copying. The information is going to be copied into something called messenger RNA. And then the messenger RNA is going to be used to make protein. And things happen pretty much around the same time. As soon as the RNA comes out from making a copy of the DNA, then the ribosomes join in and they start making protein. But in eukaryotes, it's a little different because there are membrane-bound organelles, and the DNA is kind of stored and stocked away in a membrane-bound organelle of itself, which we call it the nucleus. And the first step of copying the information from DNA into RNA happens in the nucleus. And there are steps. So first there is a pre-messenger RNA is made, and then which matures into a messenger RNA, the final product that we called here messenger RNA. And then that messenger RNA leaves the nucleus, and converting the information from RNA into protein happens in a separate compartment, which is the cytoplasm. So in terms of complexity and details, they're pretty different. So going forward, I'm going to focus on gene expression, the central dogma in eukaryotic cells, and maybe also in some steps mention what happens in prokaryotic cells. So here it is, another way of showing the central dogma or gene expression. We're going from DNA to RNA to protein. Conversion of DNA to RNA is called transcription, and conversion of RNA information into protein is called translation. And I'm going to talk more detail about this later on. So here's a GIF of showing how this works. So we'll start with transcription, copying the information from DNA into RNA. Notice this happens in nucleus, that's in eukaryotic cells. And you also notice that only one of the strands is being copied, the other one is not copied. So we call this strand that is being copied the template strand. So now the RNA is made and it's going to move into the cytoplasm, and then the components of translation, which include ribosomes and transfer RNA, come in, and they start reading the information in the RNA, and they put together a protein. And we're going to talk more about how this happens in later videos that I make after this. So then once the protein is made, then messenger moves to another ribosome, and then the protein folds, and there you go. You go from gene, which is at this step. This is the information on the gene on the DNA, and it's converted to RNA, and then that information from RNA is converted into protein. So based on what we just talked about, you should be able to answer this question, and we can talk about this if you have any questions about it during our office hours.